Good morning and welcome. My name is Hallie Harriman and I am part of the Northeast Risk Leadership Team. I'm also part of the Invasive Species Collaborative down at Virginia Tech, and I'm going to be your host this morning. A few housekeeping items before we kick off this coffee talk. Number one, please keep yourself muted for the presentation portion of the event. We will have time for questions towards the end of our discussion today, so just keep those in your pocket ready to go. Also, you'll note that this presentation is being recorded and it's going to be posted on the Northeast Risk and Northeast CASC websites once that link becomes available. So looking at our agenda, I'm going to give you some quick background slides on the Northeast Risk and the Northeast CASC, what those organizations are and what they do. We'll have our presentation and tool demo from our presenters. We'll have a quick Q&A, and then finally we'll wrap it up with some final remarks and then also a quick survey at the end. So today our presentation is co-hosted actually with the Northeast Risk and the Northeast CASC. First off, we're going to talk about the risk. And so the Northeast Risk, this is an organization that aims to reduce the compounding effects of invasive species and climate change by doing several things, by synthesizing relevant science, communicating the needs of practitioners to researchers, building stronger scientist-practitioner communities, and conducting priority research. So the risk was actually born in the Northeast and was born in the Northeast, and because of invasive species practitioners' needs to understand how climate change would impact their work. So the first meeting was actually held almost 10 years ago. It's hard to believe, back in 2016, and has been growing steadily ever since. The Northeast Risk is supported by the Northeast CASC and is a partnership between the U.S. Geological Survey and the University of Massachusetts Amherst. So our partner organization for today is the Northeast Climate Adaptation Science Center, otherwise known as the Northeast CASC. The mission of the CASC is to help fish, wildlife, ecosystems, and cultural resources adapt to a changing climate. So much like the Northeast Risk, the Northeast CASC does focus on bridging that gap between science and management to really identify critical climate adaptation challenges and to address them collaboratively. This approach involves stakeholder-driven, actionable science, as well as active engagement with partners to ensure the science meets their needs, and capacity building through networks and partnerships that facilitate knowledge sharing, much like the webinar that we are hosting today. So before we dive into our presentation, please take note of all of our contact information. Both organizations do have websites, so please feel free to visit these if you want to learn more about the projects, the partners, the research that's happening, and you can also contact us directly via email. If you're interested in learning more about our more current activities, you can absolutely join listservs by visiting our respective websites. And so now I'm really pleased to introduce Dr. Bethany Bradley and Dr. Dan Buonaiuto. So Dr. Bradley is a professor of environmental conservation at the University of Massachusetts Amherst, and Dan is an assistant professor at the University of Maryland. And today they're going to be talking to us about assisting gene flow for climate smart restoration. So with that all being said, I'm going to go ahead and turn the presentation over to Bethany and Dan. But first, I believe we have a Zoom survey. So just take 1 or 2 minutes to read these 2 questions and respond. 1, how familiar are you with the concept of assisted gene flow? And 2, how comfortable are you with the concept of assisted gene flow?
All right, so that's looking to me like we're somewhere in the middle. A couple of people have good familiarity, but most of us are sort of right in the center of kind of knowing what it means a little bit, but not having a whole lot of familiarity. Can we go on to the next one? Oh, I guess I just need to scroll down for the next one. And then how comfortable we are, right? So how familiar we are, how comfortable we are. Okay, we've got a perfect bell curve on both of these for the poll that we're all kind of right around in the middle of being familiar with it and comfortable with it. All right, so I'm going to stop that poll. I think I can close it and go on over to our presentation on this research to practice paper. So just as a reminder for folks who are either familiar or not familiar with the RISC network, our research to practice papers are syntheses of a review of the state of the science. So this research to practice paper is co-authored by me, as well as Eve Bury at the New York Botanical Gardens, Matt Brinka at New York State Parks, and Grace Gutierrez, who's at UMass Amherst. Before we get started, it's good to start with some definitions. Managed relocation as a general term is also called assisted migration. So managed relocation or assisted migration, our definition of that is the intentional introduction of either genotypes or species outside of their historical ranges into more climatically favorable regions. And there are kind of two main components of this. There's the movement of genotypes, and then there's the movement of species. So if you imagine that the green blob in this slide is the range of some given plant species, and the trees are populations of that same species distributed throughout the range, then you can imagine that if there's a warm margin to the south and a cold margin to the north of that range, that the populations of those same tree species exist across this climatic gradient, right, of warm to cold. So one part of managed relocation involves the introduction of those warmer adapted genotypes into cooler climates, but still staying within that historical range. So we're still within the green blob, and what we're doing here is moving warm adapted trees slightly to the north. That's termed assisted gene flow, and that's what we're going to focus on here in this presentation. Another part of managed relocation involves moving species outside of their historical range. So that's often termed assisted species migration. And in this case, the blue tree at the north, if we're moving it outside of that historical range margin, that would be an example of species migration. And of course, what we're concerned about is that we'll see population declines and even local extinctions at range margins, and actually not just at range margins, potentially in the middle of species ranges, kind of represented by these brown trees. And that may happen because climate conditions have become too hot, or because they've become too wet or too dry, as the case may be related to their tolerance levels. So today we're focused just on the assisted gene flow part, the movement of warm adapted genes within a population, and not on the assisted species migration piece of movement of species out of their historic native range. But our plan is to get to that second one in our next, in a future research to practice paper, a future synthesis. Okay, so before we can talk about approaches for assisted gene flow, we need to first think about how far species need to move just to keep up with climate change. So this is a scatter plot on the left of points that are from across the Northeast. So everywhere from Virginia up to Maine. And then those, any random point was then separated by a random distance north or south of up to 300 miles. So that's why you get the 300 miles across the x-axis. And then we measured the difference in temperature between those 2 points. And it's not a perfect line, but it's a pretty good correlation between distance and temperature. And I put these in miles and in Fahrenheit because it's more intuitive for me to understand, and I am guessing that hopefully that's more intuitive for most of you to understand as well. So our assumption is that plants are adapted to their current location. So for example, I'm sitting in Amherst, Massachusetts, which is represented by the star on the map on the right in Western Mass. So the thinking is, right, that if you find plants that are in Amherst, Massachusetts, and have been there for a while, then they're probably adapted to the historical climate of Amherst, Massachusetts. Another way to look at this is in a graph of temperature over time. So this is a graph from NOAA that NOAA put together for all of the different states. So if you focus on the orange line, this is actually the graph for Massachusetts. And maybe it should say I am here. So all of you who are in Massachusetts, you are there in that orange line. So you can see that temperatures for Massachusetts have already risen by about 2.5 degrees Fahrenheit over the last 50 or so years. And if you're a little bit further north of Massachusetts, so if you're in northern New York, Vermont, New Hampshire, Maine, then that temperature has probably risen a little bit more. So that's where we get up to about 3 degrees. And if you're a little bit south, if you're down in Virginia or the mid-Atlantic, then you're probably closer to that 2-degree mark. So 2 to 3 degrees Fahrenheit is what we've already experienced. And on average, that takes us a distance of about 80 to 120 miles south. So from Amherst, that is about to Port Jefferson on Long Island. So the species that are living and have adapted and evolved in Port Jefferson on Long Island are actually better acclimated to the temperatures that we're experiencing in Amherst today. And projected warming, so this is for the end of this century, so 2080 to 2100, certainly for long-lived plants, if we're thinking about shrubs and trees or thinking about populations of plants for a long time period, that's a timeline that we want to be thinking about. So on average, if you are going to about 5 to 7 degrees Fahrenheit, which is a projection for the end of this century, that takes us about 200 to 300 miles south. And for Amherst, that gets us all the way down to Atlantic City, which is kind of exciting. I've never actually been to Atlantic City, so it seems exciting. Another way of expressing this change in temperature versus distance is in terms of what climate scientists call the velocity of climate change. So this is a map of that same phenomenon, velocity of climate change, from about 10 years ago. And what it shows is that on average, another way to think about this is that plants need to shift their ranges by about 3.25 kilometers every year. That's just to keep up with climate change. And it's kind of hard to see on this image. You have to squint at the Northeast a little bit, but much of the Northeast is in shades of yellow. And those shades of yellow are actually even higher than that average 3.25. As you go further north, you have to move further because climate is warming even faster. So probably for the Northeast, we're actually looking at, maybe upwards of 5 or higher kilometers per year that things need to move. Sorry to be shifting back and forth between kilometers and miles. So then the question is, how well are species keeping up with this 3.25 kilometers per year? So this is a graph that shows the average rate of range expansion. This is for 247 native species that have been measured around the world in terms of rates of spread. So, we have some location where they were historically, and then a location where they were more recently, and can measure rate of spread in terms of how far they've gone over that passage of years. And these data were compiled by Rubenstein et al. And the average velocity, so this is for all taxa, not just plants, average rate is 1.74 kilometers per year. So that's definitely not keeping up with climate change for all of our native species around the world, but at least they're kind of moving, they're like maybe still in the race. So in contrast, if we look at just plants, and our data here are for 34 dicot species and 4 monocot species. So if we think of this in terms of that race of species trying to keep up with climate change, then plants are basically face planting at the starting gate, right? So that means that plants really need our help in order to keep up with climate change, right? To minimize that risk of population declines and local extinctions of species, especially at those southern range margins. So here's where we get assisted gene flow. This is the, again, we're gonna look at this as a means for addressing this problem of, how do we get species to move and keep up with climate change? Moving genotypes within their ranges from warm adapted locations to cooler locations may be a solution to that. All right, so I just wanted to start with some background on what we know from the scientific literature about assisted gene flow. So the first is that assisted gene flow is recommended for conservation. So these are some quotes from a paper by Millar et al. Almost 20 years ago at this point. This was the first or near the first of many academic papers that say that this type of action is needed and necessary in the face of rapid climate change. This paper was specifically focused on forests, but it's relevant to any ecosystem. And so since at least 2007, conservation biologists have been saying that we need to assist the transitions of species and in particular expand genetic diversity guidelines to be able to move warm adapted genotypes into cooler locations. And then second, this is a paper by Gann and others. It's actually a giant book practically of standards for ecological restoration. This has been updated a number of times over time, most recently in 2019. So I'll read this to you because it's a little, they're like slightly a little jargony, I think, on this one. So what this says is that reference models, so reference models in this case is what restoration ecologists refer to as what you're trying to restore to. So whatever we're trying to restore to, that model should be developed with an explicit focus on understanding temporal dynamics, so that is climate change, things that are changing over time, to develop feasible and relevant restoration designs that allow species to recover, adapt, evolve, and reassemble. And so all of that slightly jargony speak basically means that we can't use historical genotypes, species, or communities as a reference for restoration in the context of climate change. So that is no longer an appropriate reference model in many cases, maybe most cases. And then also assisted gene flow is recommended for management. So in 2023, the US Fish and Wildlife Service updated the rules associated with the Endangered Species Act to allow specifically for species to be restored not only within their historical range, but also into new ranges that would account for climate change. All right, so hopefully that convinces you that there's a lot of scientific and management backing for this practice. And so the next question is, how do we actually do this? So these are some slides that the figures from here on out mostly come from our research to practice paper. So you can also find them there and reference the text that's in there, which will hopefully align with what I'm saying too. So in our synthesis, we focused on 3 options for sourcing seeds for conservation or for restoration. So that is local provenancing, admixture provenancing, and climate adjusted provenancing. So probably the most common ones. Let's go through those. All right, so local provenancing. This means that seeds are sourced locally, so within a geographic or ecological region close to, whatever close means, the restoration site. So this is essentially how restoration has historically been done or would historically have been recommended in the absence of rapid climate change. Second major option is called admixture provenancing. So this is one where seeds are sourced both locally and from more distant regions in all directions. So you can see the little dots on the map on the left indicate locations where we would be sourcing seeds, and those include both in cooler northerly directions as well as in warmer southerly directions, but all still within that green range of the species. So admixture provenancing tends to be a good approach if we know that things are changing, but we don't really understand the direction of the change. This might be, for example, a location where we're not having as much direct effect of climate change, but there's a lot of, land cover change or other disturbance happening within an ecosystem. And then third, climate adjusted provenancing. So this is one where seeds are sourced both locally and from more distant regions in the direction of expected climate change. And then the other piece is that more seeds get collected in those locations that are more distant. So if you look at the map on the left, you see, a few smaller seed collection locations closer to the restoration site in the star, and then bigger dots, more intensive seed collection as you go further south into what should be warm adapted populations of that species. So this approach is recommended when the environment is changing and we have a pretty good idea of the direction of that change, like for climate change. So each of these options, local, admixture, climate adjusted, has a place in a restoration or a conservation portfolio. So for example, local provenancing, if we just focus on that one, that remains the recommended choice if we know or have a pretty good sense that the target species has lots of different populations locally, has a large population or multiple large populations, doesn't appear to be declining in those populations. Optimally, we might also know that that population has high genetic diversity or high gene flow between those multiple large populations. Local provenancing also is recommended, is the main recommended choice in cases where the site is in a relatively stable environment or has low environmental disturbance. So if it's a pristine site with not a lot of disturbance and not a huge exposure to climate change, then local provenancing kind of remains the main approach to use, or recommended approach. Admixture provenancing tends to be recommended if the local populations are showing some sign of decline. So that might include information about low genetic diversity or low dispersal capability if we have that, or it might just be observations of fewer populations or fewer abundance within those populations. Importantly, so as I mentioned before, admixture provenancing makes sense if the restoration site is experiencing a lot of change, but that the direction of that change is uncertain. And so in a climate context, for example, if you have a species that is really affected, really dependent on water, right? If you've got a species that needs a lot of water and it's a little unclear whether that site is getting wetter or drier, like both of those things seem to be happening at the same time, then maybe admixture provenancing and going in a bunch of different directions is the better approach for that. So then finally, climate adjusted provenancing, also recommended similar to admixture if the local population is showing some sign of decline, or if we have information about low genetic diversity or low dispersal capabilities. And then in this particular case, the restoration site is experiencing still a lot of environmental change, but that we're pretty confident about the direction of that change. So, we know that we're getting warmer and we have a pretty good sense that this species or this community is affected by temperatures. All right, and then the next question is, okay, so that's sort of the layout of the state of recommendations for assisted gene flow. The next question is, what do we actually know about examples of studies that have done this, that have moved a species from a warm adapted location to a cooler location and compared, how well those species did and if the warm adapted did better or the cool adapted did better. I'm going to show you some examples of the only study that we can find that has been published for any species that are relevant in the eastern US. And this is a study that was done in Illinois. You can kind of see the star there next to Lake Michigan. That's where a common garden site was. And then the dots that are across these, the colored bars are climate gradients. So there are some dots down in the orange where they collected seeds, and there's some dots up in the blue where they collected seeds, and then some ones that are local to that star next to Lake Michigan where they collected seeds for these three different species. It's also worth mentioning that this is a super small field, this field of experiments done to test whether assisted gene flow is effective or not. We found only about half a dozen studies that compare plant performance for local provenancing versus for climate adjusted provenancing. So here's an example of that, which is typical of the other handful of studies that we found as well. So what this paper by Woolridge and others found is that for Allium cernum, which is nodding onion, the plants that had the highest overall fitness, so the y-axis is fitness, and then across the x-axis is the source of the seeds. So the northern seeds were presumably more cold adapted, local seeds locally adapted, and southern seeds more warm adapted. So for this species, the overall fitness was actually highest in the cooler and northern populations, in which case, for to keep going with this species, actually the admixture provenancing, where you're grabbing seeds from all over because we're not really sure, which is most adaptable, would be best. For partridge pea, the opposite was true. So the plants with the highest overall fitness were from the southern warm adapted source, and the northern adapted cold adapted ones did the worst. And so that suggests that actually the best approach for conservation of this species would use climate adjusted provenancing. And then lastly, for black-eyed Susans, so these ones didn't actually have a northern seed source, or it failed or something in the experiment. In this one, the local population actually had the highest overall fitness. And so this would suggest, if we had all this information for all species, that we should be using local provenancing. So we have 3 species in this experiment with 3 different results. And as you might guess, that's really not enough to build general guidelines for what we should be doing across all of the species across the Northeast. Those half a dozen other studies that we found, which were largely from the Western US and from Europe, showed similar lack of consensus about which sources of seeds had higher fitness or higher numbers of flowers or fruits or what have you. So what this means is that we really need experiments to know what to do. This is a recent paper by NATO et al. From just last year, and it calls for restoration sites that build adaptation experiments. And it also includes a nice example and protocol from a common garden that they've created at Cadillac Mountain up in Maine. So the map on here, for example, Cadillac Mountain kind of fades into the coast of Maine, but it's kind of in eastern part of the coast, the little yellow dot. And then all of those other dots are places where they collected seed for this particular experiment. So the take-home point on this is that given that we have a lot of restoration sites, and especially for invasive species managers who are in the audience, we're doing a lot of treatments of already invaded, already disturbed sites, and that creates opportunities for restoration. Those opportunities for restoration are also places where we can be conducting adaptation experiments like this, because we very desperately need more than 3 species to help guide, which ones are going to be able to adapt in place, which ones are we really going to need to help out to move across our landscapes. So we suspect that a lot of this work may already be happening outside of what's available in scientific publications. So this is a QR code, which you can either grab now, or you can certainly grab it from the Research to Practice paper. It's at the end of that. What we're aiming to do with both the Climate Adaptation Science Center and through RISC is to try to compile information about who is doing assisted gene flow, what and to what extent we're doing that across the landscape. And that's whether or not there's any associated like science experiment associated with it, just if species, like species are being moved as part of a conservation or restoration strategy is also something that we're really interested in. So you can find that QR code link at the end of the research to practice paper as well. And we'd really appreciate it if you can fill that out if you know of assisted gene flow studies or assisted migration studies, and can share it with other folks who are doing the same. All right, and then the last thing that we have in this research to practice paper is that we've created a tool, or I should say Dan has created a tool, along with Kabira Singh, who's another member of the RISC network, that will help to identify locations where you could potentially source seeds to support climate adjusted provenancing. And so I'm going to stop sharing and turn it over to Dan to give us a demo of that tool.
Okay, thanks, Bethany. Oh no, let's see. I'm gonna try to see if my sharing works. Oh no. Oh, Bethany, I might need you to actually share for me because my new computer doesn't let me.
Sounds good. Let me.
Okay.
It's gonna take me.
Yeah.
Do you
You
want me
could just do it in the presentation.
Okay, I will do that then.
Great. Thanks.
So just showing slides.
Yeah, that's fine. So this is actually probably good because and so our climate adjusted provincing tool, right now the server, there seems like there's something going on with the server and it was crashing a little bit. So I can walk you through this tool based on uh, just based on screenshots that I happened to grab yesterday. Um, that I intentionally grabbed yesterday because I had a feeling we might be having some internet trouble. But anyway, so this is the climate adjusted provincing tool. And the idea behind this tool is that there are many great data sets out there sort of in the literature that one could use to kind of figure out where species are and where they're doing well. But those aren't necessarily the most accessible for folks who don't have time to sort of run through the literature and then code up their own, guidelines. So we put together this portal from a data set that was really compiled by Lais Petri and a group of other collaborators, where they took what it came out to about 78,000 vegetative survey plots from all around the United States, from different federal and state agencies. And they basically got them to play nicely with each other. So we have this data set of species abundance at sites from all across the United States. And so this is a tool that will kind of help people navigate this. So what you're seeing here is the interface. And the way it works is that you would put in your location in that first box, so the coordinates of your lat long. So I have it set to Amherst right now. And then in box 2, you would pick a climate scenario and a climatic variable filter. So the climate scenarios you could choose from are contemporary, +2 degrees or +4 degrees. And then we have a module for temperature or water deficit, so a measure of aridity essentially. So I'm showing you temperature here. So this is all the plots in that data set that have a temperature that is within 1 degree Celsius of our focal site, of which is Amherst, right? So if you wanted to use that sort of local provenancing based on climate, you can see that there's a whole chunk kind of in New England, southern New England, and then it happens to be also down the Appalachians where it's a little higher elevation. right? So that gives you your contemporary. So Bethany, you can advance the slide. And just if you want to see what that looks like, if we change it from contemporary to low, you can see, so these are now all the sites that are kind of within 2 degrees of our home site, or, um, and you can see that we get a lot more of a southern distribution down into my area in the Mid-Atlantic, Maryland and Virginia, as well as moving west. So this sort of gives us, again, like that temperature profile. So you can see kind of things. So this first map really shows this region. And if you wanted to, I don't have this, you could hover over one of these dots and it would give you the name of the site and the coordinates. So that would allow you to cross-reference it to the main database, which is publicly available. Okay, next slide. Okay, so what's cool is what we can then do is from that first map, the next panel that we see is basically a bar plot of the most common species in that climate range. So again, this is set for 2 degrees, 2 degrees warming from Amherst, and we can see that here are the species that are showing up at those plots that were in the previous map. And right now we have it mixed between all the different functional types, but let's imagine you're interested in, you're in graminoid restoration. Change the, okay, so you can unclick or click ones on the box here on the left, and you can see, so now we have a list of just the graminoid species. Right, that are common. And again, so we see pen sedge is the number one, and a bunch of other folks that show up. So what then you would be able to do is imagine you wanted to choose one of those species. So let's choose Poverty oats grass, which is that number 4 species, D'Antonia spicata. And we have another map, which again, this is kind of below in the window. And what you have now is for that particular species, you have its full distribution in green. So these are all the sites that it's found at in that species database, that database of 78,000 plots. And the purple dots are which of those plots it is found at are in sort of your climate zone of interest. So again, this is our 2 degree warming from Amherst. So if you were in Amherst and you wanted to do your climate provenancing for 2 degrees for this species, you might be looking you might be calling me up at the University of Maryland and saying, like, can we come collect out at your field sites for seed? So that's the tool. And then, of course, next slide, Bethany, there is an option to download the data so you can kind of here download the map data, which gives you the subset of all the plots from the species database that kind of fit your climate criteria. So that's a big data set that you could then go ahead and manipulate. And the species summaries are sort of just that, a summary of the table with the bar plots that tells you the most common species and how many sites they show up at. So that is an overview of the tool. I'm sorry I couldn't kind of scroll live through it, and I would sort of, I think we have a link for it going out in the chat. I would say give us, you should be able to get a feel for it now. I imagine that we need to do a little bit of cleaning up on the server end before it can be fully functional. But you can feel free to play with it, and definitely let us know if you have feedback. And hopefully you'll find it useful and or interesting.
All right, cool. That was great. Thank you so much, Dan and Bethany. So what we're going to do now is open the room for questions. So I know some folks had questions in the chat. If you're comfortable asking them out loud, you're more than welcome to do so. Otherwise, we'll look at those questions for you and read them out loud. But by all means, if you're comfortable, feel free to unmute yourself and ask our presenters some questions. We have one question. I'll go and get it started from Dave May. This is a great tool. Any chance it extends north of the border into Canada?
That is the eternal question. Unfortunately, the data set is only for the continental United States. Or for the United States, we also have we do have data in Alaska and Canada, but sorry, Alaska and Hawaii, but there is no data for Canada. So sorry, we know that's a big problem.
Thank you. We have another question from Steve Young from earlier. How do you know climate change is affecting population more than other threats? And can you rank populations to know which seed moving procedures to use?
How do we know climate change is affecting population more than other threats? Well, I guess we don't, depending on the location. We know that climate change is a major, fast change that's happening right now. I mean, certainly if your site is under threat of being paved over by a shopping mall, if those still exist, like that's a bigger threat. But I would say land use, land cover change to that level is probably, or like full on eradication of species, in my estimation, would be the only one that would rank higher than climate change as a threat to ecosystems right now. And I can say that as an invasive species biologist too. So can we rank populations to know which seed moving procedure to use? Yeah, this is why we need those experiments, basically. You know, like, I don't know that we can. I mean, I think we can guess in some cases that if the populations are being, you know, maintained and are large and healthy, that there's not a whole lot of need or motivation to be supplementing those populations. But if the populations are declining, or if they're small and you know, small populations that don't appear to be well connected, then I think those are cases where either the admixture or the climate adjusted provenancing is a good choice for, you know, essentially experiments, right? I mean, this is an experimental procedure to help us guide how we help species adapt to climate change.
One more question. We have one from Molly Hassett. Is outbreeding depression a big concern? In talking with many native plant groups about reforestation, I believe outbreeding depression is a biggest fear for many.
Yeah, and I think that's a great question. So the general guidelines that we've seen is to be, so inbreeding depression is, you know, when you've got very low genetic diversity, and that affects your population because you don't have the genetic diversity to adapt, right, to new conditions. But outbreeding depression can actually do that same thing, and that's if you get sources from too many disparate populations that are so different that like putting them together actually doesn't allow for building that adaptable genetic diversity that you're looking for. What we've read about outbreeding depression is basically that you should try to avoid. So actually from Dan's example of the climate adjusted provenancing tool, you should probably not get seeds from every single one of those purple points, or from, you know, from one purple point in Virginia and one in Pennsylvania and one in Massachusetts or something, but instead try to cluster ones that might be part of a similar population, might have some population flow between them within some of those worm adapted regions or southern regions. So yeah, both outbreeding and inbreeding depression are both concerns. And the problem is, you know, like, we're just not, the reality is we're not going to have genetics we have genetics for so few plants to begin with that we're not suddenly going to turn around and have tons of genetic information for all of the plants that we want to try to support with climate change. So that's why we need to be testing and seeing what works and what doesn't work. And then hopefully we've got some geneticists as part of this who can do some work on those species that we're moving to try to understand and make better recommendations about how far, you know, how many sources you should actually seed from.
Thank you, Bethany. We have a question from Robert. When you're talking about longer lived species such as woody stems, how do you manage exposure to lower limit temperatures that will persist for the coming years?
Yeah, No, that's a really good question. And there's a link, let's see, somebody put a link in the chat to the adaptive silviculture for climate change. So Katie put that in. That's another really nice site specific to trees. For trees, so for trees specifically, what a lot of foresters have done, and trees get moved all over the place because we're trying to focus on wood production and stuff like that. So a lot of forestry studies have focused on how far you can move trees. And the term for that is transfer distance. So basically how far you can move a tree without the tree without it being too cold or the tree being exposed to conditions now that it can't persist in. And that distance depends on the species, of course, but it seems to be anywhere like around 200 miles. I think it's miles. And there's a really nice reference by Pike and others about trees, and we have that cited and mentioned in the research to practice paper too. So basically the recommendation for trees, I think, is pretty similar to the recommendation for other species, which is go a ways but not too far, so that a couple hundred miles or less, but don't grab any trees from Florida, and they probably will be okay.
We have a follow-up question from Cooper asking, do you know of folks who are doing genetic informing assisted gene flow work? And I'm not sure if that's regionally or nationally, but just generally speaking, are you aware of folks who are working in this field?
I think we should open that question to anybody in the audience. Does anybody else, if you've got names of folks who are doing that work. I think that the Schoodic Institute, that they have some geneticists working on that experiment. I don't know.
Carrie looks like she put a link in there. And Jenica too. Let's see, we have a few more questions, maybe just one more, and then we'll head to our closing. But we have another question from Steve Young. Is climate change happening so fast that the time it takes to do the studies and get the materials too slow and outdated by the time that you do it?
Well, I think some of that depends on what our decisions are in terms of climate change for the future, right? So climate change is happening really fast. I think if we don't do anything, then we're in a worse place than we would be, even if some of our stuff is outdated. I mean, a lot of the studies for at least perennial, short-lived perennial species that have been studied in a bunch of these the handful of case studies are pretty recent. So it only takes a couple of years to do some of those experiments. So if the restoration is for, like herbaceous species or meadow or something like that in the Northeast, then I think we're in decent shape of being able to be quick about doing those studies and then responding to them. Trees are definitely a much harder. And that's why it's really fantastic to have these large scale plot studies where they have been, like the adaptive silviculture for climate change, where they have been planting a lot of different trees in a lot of different locations to try to understand broadly. I would love for us to have something like that, but for herbaceous and shrub species as well, because they're learning a lot and they're able to feed it directly into silviculture practices. And I feel like we don't know enough to feed into our restoration practices yet.
All right. And actually, I'm going to ask you one more question because I'm curious too. And this is from Bridget. Do you have any recommendations about commercial sourcing of seed versus wild seed collecting? Seed may be more accessible from southern locations, however, genetic diversity may be limited.
Yeah, and actually we have another research summary through RISC coming out in a couple of weeks, I think, that's specifically about grown seeds and how they narrow down the genetic diversity relative to even the wild population collection. So I think that's a challenge. I think that optimally wild seed collection would be the way to go. But we also have to live in the reality that that's really hard to do and takes a lot of time and expertise. And so, so personally, I have a meadow that I'm growing in the backyard, which like the RISC folks have heard me talk about ad infinitum because I can't talk about anything else. But the seeds from that came from Ernst Seeds, which is in Pennsylvania, which is a fairly good size native seed producer and nursery. And there's another one that's in New Jersey that I'm forgetting the name of that also we've used some of. So like those are sort of on the larger scale seed producers. But if others have, that would be I'd love to know the answer to like where everybody else goes for seed, because it's definitely a challenge to know provenancing from the from seed producers. Basically, most of them don't include that information. What I've liked about Ernst specifically is that they at least say where the seed was collected, so you have some sense that this is a New York population or a Virginia population or something of the seed that you're buying. And the more we can push on seed producers to tell us where seeds are from, the more feasible it will be. And thanks, Grace. So Archwild too, as seed producers.
Mhm. All right, awesome. Well, I think we're going to go ahead and transition into our wrap up, but I do want to say a huge thank you to you, Bethany and Dan, for presenting to us today. This has been really, really cool information that you're sharing. And of course, thank you to everyone who is on the call right now. I'm going to go ahead and screen share, and you should have, let's see, a little infographic for you for your viewing pleasure as we wrap up. But thank you again for joining us this morning. It's been a great talk, and the questions were wonderful. So thank you for sharing those in the chat. If you have questions that weren't answered, please feel free to reach out to Bethany and Dan. You can find them online, grab their emails, and send them a note. But before we sign off, be sure to save the date or register for these upcoming fall events. We have a lot of really cool things coming up. Several webinars, Hydrilla Manager Research webinar, extreme weather events and invasive species, and I know that one's going to focus more so on hurricanes, so that's an exciting topic. And of course, our second annual International Invasive Species and Climate Change Conference. So that's a 2-day event. It's really big, and it's all online, so it's very accessible no matter where you are. So again, please feel free to save the date and sign up for these. We should have registration links available for each of these. And as a reminder, this session was recorded, and that link will be shared by both the Northeast RISC and the Northeast CASC. And finally, please take a moment to complete the brief survey that's going to pop up as you leave the meeting. Your feedback is super important to us. It really helps us understand your needs and improve future events. So please take the time to do that if you have some. And again, thank you. And we really hope to see you at the next webinar. And I hope everyone has a great rest of their morning.